It is my pleasure, and now everyone comes the really exciting part of this course. We're looking at PET tracers and then at therapeutic radiopharmaceuticals. But first, let's tackle the PET tracers, and I have the immense pleasure to introduce to you Dr. Jan Andersson, who is currently the Director and Senior Radiochemist at Edmonton Radiopharmaceutical Centers in Alberta, and he's also an Assistant Adjunct Professor at the University of Alberta. Jan has worked in the field of radiochemistry for over 18 years. You look very young, 18 years, really? After graduating with a Master in Sciences in Chemical Engineering from Uppsala University in Sweden, he worked at Karolinska Institute in Stockholm, where he completed also his PhD in Radiochemistry and Medical Sciences in 2010 and took on the role of Head of Radiochemistry Production there for 2 years. He then moved to Canada to work as a researcher within the field of cyclotron production, separation and quality control of radiometals at University of Alberta and Edmonton, before coming to Edmonton Radiopharmaceutical Center in 2018. Ian, welcome.
Thank you very much for that nice introduction, and thank you to CANM for inviting me. So I'm gonna talk a bit about PET radiopharmaceuticals. The objective of this is to give an overview of general PET radiopharmaceutical theory, give some specific requirements for manufacturing of short-lived PET radiopharmaceuticals for clinical use, and then go into specifically FDG production and QC. And the outline kind of follows that same pattern. This first slide is basically just going through what a radiopharmaceutical is. You guys have seen this a lot throughout the day. So basically, you have a radionuclide with the physical data that is suitable for what you want to do, and you couple it to a pharmaceutical with the pharmacokinetics that is needed. In terms of PET radionuclides, there is a bunch out there. The most commonly used are fluorine-18, carbon-11, oxygen-15, and nitrogen-13. But from this list, the one thing to kind of take is the positron energy matters for the image quality. So fluorine-18 has the lowest energy, which means that it will travel the shortest distance before annihilation. So the resolution is better than specifically for like gallium-68 has quite a high positron energy. Yeah, so this is the chart of nuclides. So for the positrons, which are neutron deficient, they are kind of to the left of the line of stability, and a lot of these are produced with using a cyclotron, because you're basically adding a proton into the nucleus, and often you push out a neutron or one or more neutrons. So you make a neutron deficient nuclide, which then becomes unstable and becomes a positron emitter. So basically you have an unbalanced ratio of neutrons to protons. So so the nucleus is not stable. So you can get beta minus decay by converting neutrons to protons. Or you can get beta plus, where you convert a proton to a neutron, or you can eject an alpha particle, which I assume Svetlana will go into more in the therapy session. As I alluded to earlier, basically what the positron decay is when the and the image that we get from it is when the positron meets an electron. It annihilates and it gives out 2 photons with 511 keV opposite directions. So that is basically what we take advantage of when we do PET imaging. We do coincidence detection, and we use a computer for image reconstruction, and we get the PET image that we want. Also note that isotopes are chemically equivalent, basically, so the body cannot distinguish from a carbon-11 or a carbon-12, or a oxygen-15 from oxygen-16. So this can be used in a number of different ways. For example, microdosing. If you have a pharmaceutical that you want to know the biodistribution of, it might be a lot faster to label it with a PET radionuclide. Usually that would be carbon-11, because all pharmaceuticals have a carbon in them. You make a carbon-11 version of it, and there's way less strict safety regulations to do a phase 1 trial with a PET tracer, and then to go through a whole pharmaceutical clinical trial development. And you can get some really good data in terms of the kinetics of your pharmaceutical. You can also do blocking studies where you block a receptor with that same compound. So that's kind of what from a research point. What I'm more going to talk about today is F18 and FDG. So that's basically you substitute. Usually, what is being done is the F18 is substituted for an OH or a hydrogen. So let's focus on FDG. The first FDG PET imaging was demonstrated already in 1976 at Brookhaven. Reimbursement started in 1995 in the US. And FDG is able to image a lot of different tumors, and it's used everywhere. It is the workhorse for PET imaging. It was also nominated as Molecule of the Century in 2000. So FDG is taken into cells with a correlation to the cell's level of glycolysis. It is not completely correct to say it's a direct measure of cell glycolysis. It is a surrogate marker though, so it's mimicking the biochemical behavior of glucose. So it's transported by the glucose transporter, GLUT1 to 5, and it's phosphorylated by hexokinase to FDG6 phosphate. But there's not really any more metabolism, so that does differ from regular glucose. So it's trapped in the cell, and you get a accumulation of FDG, which is a measure of the glucose utilization of the tissue. Clinical use, it's used in cardiology, neurology, and of course a lot in oncology. So it can be used in oncology as a whole body measure of disease extent at primary and metastatic sites, as a measure of tumor grade through indication of proliferation rate, as a marker of therapy response, and as a method to determine disease relapse by differentiating tumor tissue from necrosis or scar tissue. It can also be a prognostic indicator of disease outcome or as a biochemical probe of glucose utilization in experimental oncology. So to make a PET radiopharmaceutical, we got to go through the whole thing, starting with a cyclotron, because we need the radionuclide. We need a GMP lab with hot cells and synthesizers to make whatever radiopharmaceutical we're making. We need a quality control lab with different equipment, such as chromatographic systems and things like that. We need QA and a lot of documentation if this is a clinical radiopharmaceutical. And then, of course, we need the nuke med department to actually administer the radiopharmaceutical and do the imaging. This is a kind of an example of a production schedule where if we irradiate for 1 hour, we get a lot of F-18. The actual chemistry takes about just under half an hour, and the quality control about 20 minutes. We in Edmonton, we do 5 batches of FDG a day, so we can of course start the irradiation of for the second batch right after the initial irradiation. So it ends up being about 1 hour in between the first and second shipment of FDG. Okay, so a little bit about the cyclotron. This is our cyclotron we have at the Cross Cancer Institute. So the major parts are the ion source and the main magnet and the targets, which are here. There are shields, so we can't actually see the targets in this picture. In addition to that, there are a lot of water cooling. All the magnets, all the subsystems have to be cooled. Everything is under vacuum, so we have I think it's 4 different vacuum pumps to keep the vacuum at a very low pressure. So basically what we're doing is we're generating a stream of particles which have sufficient energy to interact with the nucleus. And it's a charged particle with a mass. So that means it's going to be repelled by the nucleus. So we have to, it has to be accelerated to a very high speed to actually interact with the nucleus. So I think we're talking about about 20% of the speed of light. So this is basically what a cyclotron is. It's a big magnet, and you have an alternating current. So the particles moving in a circular orbit in the magnetic field, and this means that smaller force can be used, but it's many times to get a high power machine. So the Ds are basically, it's a RF system which actually it's hydride ions that is being accelerated, so it's attracted to the leading edge and repelled by the trailing edge of the Ds. And when the hydride ion has been accelerated enough, we basically have an extraction foil. It's a carbon foil that takes away the electrons from the hydride ion, makes it a proton, which means that it has then the opposite charge, so it goes out to the target. It's repelled from the magnet. And this is a target station. Can't really see too much about it from this picture, except that there's a lot of water cooling going on. Like it gets hot. So the targets, they can be gas, liquid, or solid. The most typical gas reaction is nitrogen-14. You irradiate with a proton, an alpha particle is ejected, and you make carbon-11. It can be a solid. The example on the picture here is boron-11 PN reaction to carbon-11. That is, to my knowledge, never used anymore, but other solid targets are getting more used, like the gallium-68 from zinc, or copper-64 from nickel or zinc, and zirconium-89. So there are, I could see in the future it's going to be more and more solid targets. The hard part about that is the actual transfer of the solid target into a hot cell, where you can do the manipulation. With a gas or a liquid, it's easy to have transfer lines. So the liquid target is by far the most common. And so to make F18, we start with O18, we radiate with a proton, it ejects a neutron, we get F18. So in our center, we get the F18 from a target station, we download the F18 in water into the hot cell. We actually split it up in the hot cell if we need to do different radiopharmaceuticals or research, and then whatever F18 we need for clinical use, we send up to our hot cells. For FDG, a little bit of a history. It started with electrophilic fluorination, so using F2. It is a mess to work with. F2 is very reactive. It's a mess to get out of the target, and it becomes very low specific activity product. So instead, what is done is nucleophilic fluorination with F-. So no carrier added F-, fluorinating F- can be produced. A lot higher amounts radioactivity yields is available. But F- is not a very good nucleophile. So this is basically the reaction scheme to make FTG. You start with mannose triflate. You have F- in cryptofix. You do hydrolysis of the protective groups, either acid or base, and you get FTG. So it started with the acid hydrolysis. It was Hamacher who was the first to publish about this. And then Furchner used the sodium hydroxide, so the base hydrolysis. The difference there is the base hydrolysis takes 1 minute at no elevated temperature but the acid hydrolysis, it's at high temperature for a long time, so you actually gain yield just by the shortening of the production process, because you don't lose activity through to the half-life of F18. And again, F- is not a a great nucleophile, and it's not very reactive in water. So the first step is always to get rid of the water that is from the cyclotron. So you have F18 in usually 3 to 4 milliliters of water. You trap the F18 on a QMA cartridge or an anion cartridge. So most of the water goes right through, and then you can elute it with a cryptofix acetonitrile potassium carbonate. So then you get also a potassium as your cation, and that can be caged by cryptofix. So you basically get a very large diffuse cation to your fluoride. So that makes the fluoride more reactive. There are some other cations that can be used, especially TBA, tetrabutyl ammonium, is used quite a bit these days. But that is one of the things to think about when doing fluorine-18 chemistry. The second thing is to get rid of the water. There's still some water in the reaction vessel after eluting it from the QMA. So we use azeotropic drying. So basically we use the fact that acetonitrile water makes an azeotrope. So the boiling point of this azeotrope is significantly lower than just water. So after we have eluted the F18 and the water is evaporated, we do successive evaporations to make sure it's dry, and all the components is also rinsed with acetonitrile to get rid of moisture. So what we then have is the fluorotetraacetyl glucose, the protected FDG. So we do base hydrolysis to get to our FDG. I think everyone is doing base hydrolysis these days. If you do acid hydrolysis, you could end up with the chlorine substitution. And FDG does not really have to be separated from the precursors, and since it's just a very small amount of benign sugars. So these are the anomers of the fluorosugar. So you could have the alpha glucose or the beta glucose, which is the FDG, or the alpha mannose or the beta mannose, which is FDM. So in North America, we don't check for FDM. In Europe, they have to do a HPLC analysis to check the amount of FDM in their product. Now a bit of a history of the synthesis units. This is the first that was used in Edmonton in early 2000s. The G Tracerlab FX, which is a fixed tubing synthesis unit. It works great. You can do a lot of different chemistries with it, but for routine clinical production, it has to be washed and rinsed in a good manner so that you're sure that there's nothing remaining in the tubing between batch to batch. It's also it has two, it's a double run box, so you actually have two. It's like having two ASUs in one hot cell. But yeah, you have to you have to clean the tubing, and radiation damage on the valves and tubing can be quite extensive. And then we had the TracerLab MX, which was from the beginning the coincidence technologies. It was the first really cassette-based synthesis unit. So it's a sterile cassette. Everything that touches the product is in that cassette. So you remove it and then you put a new one on for the next run. And yeah, it makes the regulatory compliance quite a lot easier. And it's also easier to operate. At the same time as this, the FDG yields in the last 20 years have gone up, or the F18 yields have gone up significantly. I think in the beginning of the 2000s, a lot of people still used silver targets, which are not the greatest. Now the industry standard is niobium targets. So from 2008 to 2016, it's the targetry improvements have been significant. It's not uncommon to produce 30 curies of F18, and some big centers in the US actually make 20 curies of FDG in one run. These are the synthesis units that are out there today. There might be more, or I know there are more. These are the four big ones. The Neptis, the Traces, and the GE Fast Lab all three are kind of based on that coincidence unit that was made in 2001 or so. Just different versions of it. This IBA Synthera is slightly different. The Traces units is the one that we're using in Edmonton. The good part about it is we actually can make 4 runs on one cassette. So it's the FDG quad. So we load it at the beginning of the day, and we can do 4 out of the 5 batches on the one cassette. Synthera has a similar thing where they actually where you remotely can switch cassettes on the unit. It's an automatic cassette switching. So multiple separate runs is now possible when the unit is loaded in the morning. We have a potential of a huge output of FDG. So once we have the FDG produced, we need to have quality control over it. I don't know if, so these are the tests we do. We have in appearance, so visual inspection, that it's a clear, colorless, and there's no visible matter in the product. We have the assay, the total radioactivity, radiochemical purity, which is we do by TLC. Radiochemical identity, again by TLC. We measure pH using pH strips. We have to do filter integrity of our sterile filter. So after the filtration takes place, we put a nitrogen pressure on it to see at what point we basically break the filter. And each filter has a specification on how much pressure it's supposed to take. We do a radionuclidic identity using half-life measurement. So we basically make sure the half-life is somewhere between 105 and 115 minutes. We do a chemical purity test for cryptofix. We do bacteria endotoxin tests, and Charles River now have these rapid tests. Takes about 15 minutes. I am hearing so it's based on LAL, which we get from some sort of what is it? Crawfish? No. Horseshoe crabs, that's what it is. So I'm hearing those are not doing well, so I don't know how long this is going to be going for, but we'll see. We do residual solvents for acetonitrile and ethanol, and it might also be acetone if that is used in the process. We do radionuclidic purity at least annually to check our F18 that it's pure enough, and sterility, which is the only really post-release test for our FDG, because it takes 1 to 2 weeks to do sterility testing depending on your method. Before we can start using it as a clinical product, though, we do need to check the stability of the FDG. So for each ASU, we do have to make sure that well, we basically do a production, regular quality control, and then we do a stability study at the end of whatever shelf life we want to put on our product. So for FDG, it's usually 12 hours. And we need to then also check the container compatibility. So we do it upright, inverted, to make sure the stopper or anything in the container doesn't react with our radiopharmaceutical. And we also check the integrity of the container closure. If we're shipping, we also, the radiopharmaceutical, we do have to do a shipping study. So basically it's the same thing as the stability study, but we do have to send it out, at least do a pretend shipping. So if it's for Calgary from Edmonton, we send it out on a truck for 4 hours to see that nothing changes of the product. And this should ideally be done at kind of the worst case scenarios in terms of temperature. So in Edmonton, you could envision -30 or +40, but it's really hard to hit those hit those levels, or hit the date to do it. These are some packaging. The green ones are what we are actually using in Edmonton, which is more of a homemade kind of a thing that we have validated from a CNSC point, but there are also commercially available, the Intego or the Comisher packaging systems. The problem we face with those is that they're actually not, they don't go up to the activities that we are shipping out. So I think they're graded to 80 gigs or so. So again, we are shipping to Calgary, not so much to Winnipeg anymore. And the decay really plays a role there. If we start with over 100 gigs after, and Calgary gets it after 4 hours, we're down to below 40 gigs that they actually receive and can use in their clinic. And again, so if we actually start with 250 gigabecquerels of F18, we make 136. They could make 9 patients, and they have about a gig left at the end of it. So let's say we want to make a lot more. We start with 400 and we make 220 gigs of FDG. We might add 1 or potentially 2 more patients. It doesn't scale up due to the decay. There are also some transportation issues if you want to use a commercial airline. It will sit at the plane a long time before it actually arrives or starts going, because due to their TDG requirements at the commercial airlines. So what we do is we do ground transportation wherever possible. I know actually hiring your own airplane is something people are doing. So it's an option to consider as well. So just a little bit about fluorine-18. Some other radiochemistry. There's a bunch of different things that can be done. I'm not going to go through it all. It's also possible to radiolabel peptides with F18. For example, the CIFA chemistry was developed by Professor Schirmacher at the University of Alberta, and he was also here in Montreal before. And later on, I'm gonna show an example of the aluminum fluoride complex as well. And this is just a list of some of the most used PET radiopharmaceuticals right now. I'm not going to go through the list. I think the slide deck will be sent out after. So one of them is F-DOPA. And there you have on the top is what we actually do in Edmonton. I'm not going to go into any detail at all, other than just say it's basically 5-step reaction after we get the fluorine-18 on the incorporated, which is a lot. That's not usually what you want to do. You want to work fast. You want to incorporate the F18 at the latest step as possible. So recently, Sarat et al. Actually made a method work with a copper mediated reaction where it's only 2 steps, and that's now being incorporated. So F-DOPA production is getting easier. And then for PSMA, I just want to highlight it because we started a clinical trial about a year ago, and it's really taking off. Like there's a big clinical demand for PSMA imaging. We are using the PSMA 1007. There are a couple of other options like DCF-PEEL, which is a fluorine-18 labeled PSMA agent as well. And then you have gallium-68 PSMA 11, which I think is close to being approved. And for these type of products, so like the non-FDG radiopharmaceuticals, the complexity in quality control, it steps up a bit because it's not as straightforward. Specifically, we do need to measure the actual chemicals in the product, like the cold PSMA and the precursor, if there's anything still in the product. So we use HPLC analysis for that. So it's to be able to do that, you do need a different kind of infrastructure than just a regular FDG production site. And also we have the gallium dotate here, where you, if you do it using a generator, you do need to test for the radionuclidic purity at least periodically, so to check that the germanium-68, that you don't have breakthrough in your generator. And it's I guess the gallium, it's coming more and more, and I can see why, because of with the generator and the kit, it can become more like preparation and not manufacturing, which is a lot easier from a regulatory standpoint on the site doing it. As a radiochemist, it hurts me because it's so inefficient. You end up with from one production, you can do 1 or 2 patients. It's very inefficient, but I can see the value of it too. And then just want to put this up here for as FOPI was the SNMMI image of the year in 2022. So this is actually one of those aluminum fluoride complex chemistries. So then just a short comment on the access to PET radiopharmaceuticals in Canada. So this is the list of what is approved. So in Canada we have FDG, sodium fluoride, fluorbetabane, and gallium-68 dotate. We have the cold kit, and I think one of them is the actual solution. So even at that, because of the long distances within Canada, except for FDG, nothing is really available in Western Canada, because it's all the market authorization holders here are all in the eastern, on the eastern side. And if we then compare it to the US, the FDA has approved quite a lot more, and I don't think it's easier to get approved in the US. It's more of a more of the business case is not worth it here in Canada. So, but I don't have a solution. It's just, it is a problem. Thank you for attention.
So we have, is this working? Yeah, we have time for a couple of questions. I have a question, Jan. Please. Can you come up here? Or just grab a microphone anywhere, because the people can't hear.
Hi, it's Joseph again from Timmins. I work in a very kind of remote part of Northern Ontario. What type of patient load do you think you need to have per day to support PET imaging? Just we have a lot of pathology in our part of Northern Ontario, and I've been asked before I came, what would a PET scanner be feasible? But I was kind of wondering, with the cost of dosing, independent of the camera, just the cost of the dosing, how many patients would I have to do a day?
Oh, I don't know if I have an answer for it. Usually like a fluorine-18 radiopharmaceutical that we produce, the cost is, it really depends, but let's say ballpark $2,000 But then, so that is per production.
Yeah.
So if you do more patients, the cost per patient goes down. I don't know if that would scale if you have very low patient numbers though, because a lot of it is like service contracts and stuff like that. So
Yeah, I know there's a lot of logistics with delivery and you know, fee schedule and what Cancer Care Ontario covers. So yeah, it's not an easy question, but you know, some of the operations in Calgary, let's say, how many patients they do at a clinic basis to make it viable for them.
I believe so. Calgary buys from us, and I think they have 2 cameras, and they do like 20 patients a day, maybe more.
Yeah, that's great.
Still have time for a question or two?
Hi, sorry to interrupt. There was a question online about rubidium-82. And I did mention that rubidium-82 is also approved in Canada commercially.
Oh yeah, that's right. Okay,